Let's take a look at what you're doing this week. The topic is estimating species richness. You have your overview, you have your learning objectives. So it talks about species accumulation curves, determining what kind of sample size you're going to need to get an accurate estimate of species diversity. And then you'll also be working on classifying organisms, and you'll go over some safe laboratory practices. One of the first things you want to do is look at these two guidelines here. One is on lab safety. The other looks at how to plan for a trip to a national park. And then you also have a link on how to grow bacteria. And then, of course, you have your weekly lecture over here. Your first assignment will ask you some questions about going over that safety material. Here it's asking you, considering your assignments, what kind of safety measures will you use? You'll put that into your assignment Dropbox, and down below we see the grading rubric for that. Your second assignment is to create a species accumulation curve. It shows an example of a species accumulation curve. And basically what it means is if you go out and you start sampling, if you only sample a small number of locations, so let's say you have a big field of wildflowers, but you only sample a very small portion of those, then you're going to get just a few species. And take another sample and then another sample and another sample. The more samples you take, the more new species you'll acquire. When you take two samples, if you have two of the same species, you don't count those. You only count the new species for each time here. But every time you increase your sample size, you'll probably get some more species. But at some point, that's going to start to level off. Because at some point, if you've sampled a lot, you're going to just be resampling the same species. And that's what happens when it gets flat up here. You're sampling over and over again, but you're not getting any more new species because essentially you've sampled intensely enough to get them all. When you're looking at a study and you want to know how much do I really need to sample, you can look at these curves to decide whether you think you've got a good enough sample that's an accurate representation of all the species that are there. These curves here that continue to go up, we haven't sampled enough yet because it's still going up. But when they flattened off, we have sampled enough. So this is looking at those species accumulation curves. So whenever you're going into an area and want to estimate species diversity, it's important to take a look at these species accumulation curves to get an idea for what kind of sample size you're going to need. So it's asking you to go out and find an area where there's a bird feeder, or maybe there's an area where birds congregate near you. Maybe it isn't a bird feeder, maybe it's a pond or something. So if you can find an area where you're going to see a variety of different birds, you want to use that area. So if you have a bird feeder in your yard, you can use that. You can use other areas. Again, some park areas will have a lot of species that come to them. Maybe not so much because there's a feeder, but because there's ample other resources there. If you really can't get out, there are a couple of different webcams that you could use for this, but it's a lot more fun to get out and look at them in real life. So what you're going to do is you're going to watch this area for 2-minute increments. So over this 2-minute period, how many species do you see? And then you're going to do it again. Over the next 2-minute period, how many species do you see? And then of those two samples, how many were new species? So when we take our two samples, we don't want to add the overlap of species. We only want to include now the new ones. In this data table here, your first sample, this is your first 2-minute period. How many species did you observe? Well, of course, the first sample, they're all new. But in the second one, you're going to see how many total did you observe, and then subtract out any overlap that you had, only the number of new ones. And then over here, you're going to look at the cumulative number. So if I had 5 here in my first sample, and then 3 in my second sample, my cumulative number over here is now 8, because I have a total of 8 that I've seen so far. So what you want to do then is to create a graph that looks like this. So you're going to have the number of samples, these are your 2-minute time periods, and the total number of species richness. And you're going to try to see where this graph that you're creating levels off. As it says here, you want to continue sampling until it's leveled off for at least 3 samples. So if you're still finding new species, then you want to keep going until you get essentially 3 samples in a row where you're not finding any new species. And then you have some questions that you can answer about what you're finding and about the process here at the end. And below the instructions, you have your grading rubric. And the next part of your assignment, and you may want to start this earlier in the week before you do your species accumulation curves, you're going to set up your bacterial growth experiment. And this leads you through the bacterial growth experiment pretty well. You want to make sure that you start your data collection at least by 3 a.m. on Thursday of this week, because your final collection day will be Sunday at 3 a.m. and you'll need that much time for the bacteria to grow. So set up by Thursday, collect data on Sunday, do your write-up on Monday. So you're going to have 9 Petri dishes. Follow the instructions for creating your agar. Now, one important thing to do when you're pouring the agar and doing anything with your Petri dishes is don't leave them sitting open. Unfortunately, the one video instructions kind of has these things just laying on a table and they're open. Normally, when you watch microbiologists, they'll use what they call a clamshell technique, where they just basically kind of lift up the corner of the Petri dish and pour what's in there and then close it back down, because you don't want things from the air just falling into your Petri dish. So you're going to have 9 Petri dishes. They're all going to have agar in them. The first 3 are going to only have the agar. The second 3 are going to have the agar and a piece of blotter paper that is just blank. So you're going to soak it, but not in a cleaner solution. And then your last 3 are going to be with the agar plus the blotter paper that has been soaked in a cleaner solution. So you're trying to see how effective is this cleaner at stopping bacterial growth. Well, up here in 1 through 3, I'm not using anything, so I'm just seeing how well the bacteria grow. Here, I'm looking at what happens if I just put wet paper in there. So this is going to be a control, right? We want to control for the effect of, well, what happens if bacteria just hate blotter paper and it has nothing to do with the cleaning solution, right? So we have to control for that. We control for the presence of the blotter paper. And then this last one, we're actually doing our experiment. How effective is this solution at preventing bacterial growth? So this tells you how to process your blotter paper. You want to make sure that you're always using clean tweezers when you go from one to another. You obviously want to use a new sterile swab every time you do it. Now, when you're doing your cotton swab, you want to gently rub it across the agar. You don't want to dig into the agar. And it shows you this technique. You streak it by just kind of like rubbing it back and forth across the plate, but not digging into it. And then you turn it basically 90 degrees and you do that again. And of course, make sure you watch this video that talks to you a little bit about how to grow bacteria. It's going to have you do a cheek swab and look at the effectiveness of this cleaner solution at stopping the growth of those bacteria. So once you have your agar, your bacteria, and the ones that require the blotter paper in there, you're going to seal that with masking tape. And so what you're trying to do is to prevent the bacteria from getting out and from other things getting into your Petri dish. You'll use this Google Doc for your data collection. You can see what that data collection table looks like. You're going to take observations on day 2, 4, 6, 8, and 10, describe it, and then you're also going to count the number of colonies, and you're going to calculate the percentage of cover. It mentions right here in step 3 how you're going to calculate the percentage of cover. So you drew a grid on the top surface of your Petri dish, kind of like graph paper. So you're going to essentially ask how many grids have colonies in them, and then what is the percentage of the total number of grids. So if I have 100 grids on the top of my Petri dish, and 10 of those grids out of 100 have colonies, then I have 10% coverage. When you submit your Google Doc, make sure that it is editable to anyone with the link so that we can get into it and see it. This is all in a discussion posting. So you'll have your initial post where I did some initial stuff, and then I want to add to that. So I'm just going to reply to myself in the posting. And then at the end, you have your checklist. If you encounter any issues, you have any questions as you're going through this, please let me know, and we can try to schedule a time to meet virtually.